Thank you all for being here. I just wanted to say thank you so much to Frank and the Forever Healthy team, and to all of the people here in the audience, many of you investors, people who I've talked with over the past 5 or 6 years. I come from big data. I'm not a biologist, I'm not a chemist, more in the big data and physics space. And it took a while for me to figure out things that I needed to figure out, and you all helped. And so I just want to say thank you for that. And I'll start with the presentation. So I'm Joshua McClure. I'm CEO of Maxwell Biosciences. I'm a co-inventor of the drug discovery platform. We target pathogens and cancer. We've recently had a NIAID-sponsored study show that we are pan-coronavirus and pan-influenza in animal and in lab studies in vitro. We have been shown to be quite effective against all tested strains of enveloped viruses with one compound now, all tested strains of enveloped viruses, drug-resistant fungi, 45+ bacterial strains, biofilms, and chemotherapy-resistant cancers. So we're very well funded. We've received $35 million in grants and awards that have backed our research, mostly with independent investigators around the world, and a portion of the $66 million recently awarded NIH Antiviral Drug Discovery Fund will be coming to Maxwell Biosciences as well. In 2021, we received $10.8 million in a VC seed round, and we're currently raising $20 million this year. Forward-looking statements disclaimer. So let's talk about the history of this company. Back in 2015, when I was still in big data in the tech industry, I saw this really interesting study that came out, heterochronic parabiosis. I think most people know about that. The Conboys and others really pioneered that area. And what really just wowed me, and I'm sure wowed you, was that you see for the first time an old mammal become young. And so true rejuvenation. And this became a highly repeated study over time, and for good reason, because what we saw was this old mouse, when connected to the young mouse via these tiny little capillaries in the skin, we see this the opposite effect, right? We see it become young. And what is young? We're not talking about chronological now. We're talking about biological youth, which is, in this case, strong antimicrobial performance, low inflammation, strong apoptosis, so the tissues are being replaced, which yields strong skin and strong tissues, strong epithelial borders, which maintain homeostasis, fast stem cell recruitment, and high angiogenesis, which promotes blood flow to all of the tissues. Now, WTF, how did that happen? And wow, right? How can that happen? Well, I'm sitting here in my big data office thinking, okay, this is much bigger than anything in tech, bigger than commercial real estate. It's bigger than anything I can possibly imagine. How could this possibly be happening when these tiny little capillaries, so they're smaller than what a blood cell can go through, these tiny little capillaries. So it must be a peptide. Those are the smallest proteins in the body, the peptides. And there's thousands of them circulating. 1,000 to 3,000 different peptides are circulating in your plasma at any given time. And they're tiny. They're very, very small, smaller than the wavelength of light. So we just went into a lot of scientific data. I left my job. I got bought out by a billionaire who's the chairman of my last company, and started really looking into this. We received a lot of government grants and awards, as I said, and then there's been a lot of peer-reviewed publications over the past few years. So from 2016 to 2018, what we did was big data analysis on blood plasma. We stayed kind of under the radar, didn't really tell people much about what we were doing. I'm not going to go into the details here, but there's a system and a process, gathered a lot of data, worked with a lot of folks in this room, and essentially mined the molecules that exist naturally in the blood plasma, because we knew it had to be in the plasma. It was circulating back and forth between those mice. And you see down there at the bottom this weird thing, LL37. What is that? I don't know. It's LL37. It is low in aging folks, and it's high in youth. So let's look more into that. So I went out and started taking some classes at the Buck Institute. There was something called the MIND dementia therapy. And I started looking at correlations from the MIND dementia therapy to what we had in our big data analysis. And look at that. There's LL37, which is upregulated by these MIND therapy supplements. So what is LL37? Well, it has antimicrobial activity, stem cell recruitment, angiogenesis, wound healing, cytokine management, which is inflammation, and apoptosis. And what does that remind you of? It reminds you of that young mouse, right? So I thought, well, it's correlated in the data analysis. It's correlated to the heterochronic parabiosis. It looks like we have like triple verification here that LL37 could be a silver bullet, right? So let's look more into that. So it turns out that LL37 is naturally upregulated by vitamin D, so sunlight, good nutrition, and exercise. So everything that your grandmother told you to do to stay healthy, what you do for your children, what you try to do for your health, and maybe not all the time, especially while traveling to conferences. So that essentially results in a youthful glow. You can see when somebody hasn't exercised for a while. You can see when they haven't been outside enough and say, hey, you need to change your diet, start exercising. That's essentially LL37. It's a heat shock protein, so spas, sauna, that sort of thing upregulates LL37. It does all kinds of things all over the body. This is just a tiny little sample of the studies. There's literally thousands and thousands of scientific studies on LL37 because it is the only cathelicidin antimicrobial peptide of the human species. Many other species have these cathelicidin antimicrobial peptides, but they have many of them. Like we only have one. So it has to be essentially like a Swiss Army knife. It has to do lots of different things for us, right? It has to keep us alive. So think of LL37 as long life 37. If you don't have it, you don't have a cathelicidin antimicrobial peptide. So take your vitamin D, get exercise, get out in the sun, and play. That upregulates LL37. It does something for everything. So a possible super drug, right? Yes, I got super excited about that. Okay, I got it. I figured it out. Let's do LL37 as a drug, except you can't. What do you do? So this peptide is not druggable. That is why you have not probably heard about it from anybody pitching anything except for maybe endogenous upregulation of it, because it breaks down so quickly. This peptide is broken down by bacterial enzymes, it's broken down by fungal enzymes, and it's broken down by natural enzymes that everybody produces at totally different levels. So in one person, you're going to have LL37 breaking down in a couple of hours, and in another person, you might have LL37 breaking down in 6 minutes. So an injection from a doctor, they don't know what to dose you, right? So it's completely unpredictable in human trials and also in clinics. It would have to be personalized, not a good drug. So peptides break down due to enzymatic degradation. That's where I learned this. So enzymes cleave these functional side chains. So all the functions are here like keys attached to a side chain. You can see these sort of blue keys here. These are the side chains on a peptide. And enzymes are like scissors that just snip off those keys. So you get your keys out of your pocket and, oh, it's gone. The enzyme broke it down, and so it's essentially disintegrating all the time. Now, what if we could come up with a small molecule composition, a small molecule technology that could make floppy small molecules that fold like proteins? Wouldn't that be amazing? That is the fundamental scientific breakthrough that is going to change everything. In a few years, you will not be taking antibiotics anymore. This will change everything. So this is the claremer platform. We attach those side chains at the nitrogen, The enzymes and proteases that are produced by all of you and by bacteria and fungi are not able to recognize that connection, and if they could, it's way too strong for them to break it. So we're able to attach those functional side chains, those keys, to the nitrogen, and it's now druggable. It's processed out through the liver and through the kidneys just like a regular drug. So the claremer platform solves two key problems: peptide instability, just talked about that, and the one drug for one bug limitation that we all just assume is what everything's going to be in the future. That's not the case. We are going to have one drug for many bugs, and that's what we got here. So we have now proven biomimicry over the past few years. So LL37, that peptide I told you about that breaks down so quickly, is virucidal, fungicidal, bactericidal, and oncocidal. It has to be, or you'd be rotting in your chair right now. It is your life support system. Now, we have mimicked that life support system, synthetic life support system, a synthetic immune system. It is virucidal, fungicidal, bactericidal, and oncocidal. It's pan-coronavirus, pan-influenza, all tested strains of SARS-CoV-2, rhinovirus, hepatitis, many different resistant bacterial species, and untreatable fungal species, and then oncocidal in actual human tissues human cancers that have been transferred over into a mouse. So this is a really big news here. Fauci's team at NIAID recently funded a gold standard COVID-19 hamster study in which we were able to prevent all of the symptoms of COVID-19 in that study. So we are now redoing that study and going deeper depth into the data before going into human trials. And our portion of the $66 million that just came into the antiviral defense fund for Stanford will hopefully pay for that human trial. So it was both prophylactic, so preventative, as well as therapeutic. So imagine airline crews, retail staff, et cetera, taking a nasal spray. That's what we're going to be using to prevent the symptoms of COVID, and then also potentially for influenza and others. We also have in vivo animal efficacy against bacterial pathogens, completely wiping out a drug-resistant pathogen in mice. And this is what I was talking about before, one drug for many bugs. So Maxwell's claremers are active against all enveloped viruses, almost all of them. We haven't tested all of them, but I mean, there's thousands of them, but we believe that they are likely effective against all enveloped viruses because we target a specific lipid in viral envelopes that is not in healthy human cells. That same lipid is in all bacterial and fungal membranes as well as biofilms. And Maxwell claremers are protective against infectious disease in infected animals. We've done 3 different animals prior to human trials. And they're ideal drug candidates for multi-pathogenic diseases, right? So diseases that are complicated by bacteria and fungi, right? So post-viral syndrome is you get a virus, and then you get these fungus and bacteria that come in and opportunistically infect. Same thing in cancer patients where your immune system is depleted. So ideal for those situations where the immune system has been depleted. This is more data from the NIAID-funded work where we have incredible potency, nanomolar potency, which is tiny, tiny, tiny concentrations against SARS-1, SARS-CoV-2, human beta coronavirus OC43, and MERS, all with the same compound, and then many other viruses as well. You see influenza, avian flu, swine flu, herpes. We have a patent in process right now for an anti-herpes, anti-venereal disease condom and personal lubricant. And here's some data in vitro efficacy against many bacterial pathogens. You see here many drug candidates from our claremer platform that are effective at incredible potencies against all of those. And then you look over the side here, these are toxic. The blue compounds there that have already been approved by the FDA are the types of antibiotics that will hurt you if you take them too much. So you need probiotic therapy afterwards, and don't overdose on those. That'll hurt you, right? I bled for a week after taking Cipro. So, world first accomplishment, potent efficacy against all key biofilms, bacterial and fungal. So biofilms are one of the reasons why chronic sinusitis is chronic. You can't treat it with antibiotics because the biofilms protect that polymicrobial ecosystem, viruses, fungal, and bacterial, and create drug-resistant bacteria, fungus, and drug-resistant viruses. World first accomplishment, another one. Potent efficacy against all key fungi, mold, and yeast with a single compound. Same compounds as you saw with the bacteria, last slide. Plus one article, significantly inhibited tumor growth in a human breast cancer xenotransplantation model without any noticeable acute adverse effects in the mice. We see this over and over again. No toxicity, no inflammation, many tests. Is it a panacea? Well, let's just say that it's a $121 billion revenue target just in infectious disease without cancer. Now, how is this happening? Mechanism of action is membrane disruption. We target phosphatidylserine, which is inherited from the infected host cell when a virus buds from that cell. So the virus cannot evolve that membrane. We don't target those proteins, the proteins that we all know evolve with SARS-CoV-2. We target the unchanging membrane that your own body marks for your own immune system to destroy. So biomimicry, we're mimicking the body's immune system. So this is the destruction of the virus. We don't just inhibit the growth of a virus, we completely destroy it. And we've proven that with electron microscopy images that were provided to us by the University of Texas when they tested our drugs in their biocontainment level 3 facility against both herpes as well as SARS-CoV-2. These are the higher resolution images here. You can see well-formed SARS-CoV-2 and HSV there. You can see the very recognizable corona on the bottom there, SARS-CoV-2. And then these are partially disrupted viral membranes. They pop like a bubble. And so it took thousands of images to find that. It's almost like a piece of art. And then these are the fully disrupted images here. And that is essentially like a splat of goo there that you would see from an insect on your windshield. Compare that to the well-formed virus there. You see it's completely destroyed. And then this is with bacteria. So we don't pop the membrane of a bacteria, which is very, very important. If your own peptides did that, then the other bacteria would be able to form resistance to it, but they don't. What they do is they form a pore in the bacterial membrane and they freeze the metabolism of the membrane. It's a silent death. The other bacteria around it don't even know it died, and so they can't form resistance to it. We don't form resistance. So we tested against Pseudomonas aeruginosa, which is the most aggressive resistance-forming bacteria. And you can see here, we compared against gentamicin, and our claremers formed no resistance whatsoever, did not inform the Pseudomonas aeruginosa as to how it was being killed, versus gentamicin explodes the bacteria, and it's able to look inside and figure it out and resist. So we have also tested in many animal strains all the way up to 100 times the therapeutic concentration and still see no toxicity whatsoever. We see favorable selectivity profiles in human tissue models. So this is air-liquid interface pseudostratified epithelium of the lung, which is where we're planning to start our first trial. And what we see is no toxicity, no inflammation. Tested many different compounds in tail vein IV injection in rats. We see no adverse effect across all three of those lead candidates. We also see no adverse effects, no toxicity in highly sensitive mouse neurons. Claremers are not inflammatory and not immunogenic, genetic. So what you see sometimes in peptides is that you see the body recognizes that peptide as something foreign and then starts to inflame. What we see here is none, right? So these controls here over on the side, you see natural inflammation. We know that those inflame animals. This is an in vivo study. The animals showed zero inflammation whatsoever from our compounds. So the claremer platform advantages are no to low toxicity, high potency, strong biostability, which means they don't break down in vivo, We address a broad spectrum of unmet medical needs. They're very small, precision engineered, no development of drug resistance, long shelf stability, which means we almost certainly don't require a cold chain, which means Africa, India, many other places in the world will now be available for these compounds, and very scalable manufacturing. We own our patents outright. There's 6 issued patents. We have many others that are on the way. Our team is absolutely incredible. Dr. Ed Rudnick is in the audience today. He's the former US head of Shire Pharmaceuticals R&D department, and brought with him some of the key leaders from Shire Pharmaceuticals team. And here's our pipeline. We're going after long COVID, mostly with government money. Chronic sinusitis is our lead candidate, and we're looking at some others as well. Chronic sinusitis is a polymicrobial infection of the sinuses that is protected by a biofilm. It's suffered by about 11% of the global population, about 30 million people in the United States alone. It's a combination of fungal and bacterial pathogens as well as viruses, and it's totally different for each person. So what we had to do is go review all the literature and get the list of pathogens and then test against all of those pathogens. That's a risky proposition. The data that came back has said we're effective against all of them, including the biofilms, which is many world firsts there. So again, one drug for many bugs. That means we can scale this manufacturing-wise, scale-wise, at a level that's only seen in livestock right now. So we're talking about below $1 per patient per day for treatment. Very, very, very affordable. We're headed towards the clinic. Next year, we'll be in first-in-human trials, and on schedule and on budget for that right now. We have an existing pharma partnership. We're expecting to get partnership royalties sometime in 2023, and then we've already been talking with other major pharma companies that are very interested, especially in the chronic rhinosinusitis indication. Now, what is the company worth? Well, we don't know, but other biotech platforms that IPO'd successfully prior to the big biotech bubble that just occurred, so 2015, 2017 through 2018, we see upwards of $1 billion for preclinical and phase 1. So we're planning to IPO after that, so it's going to be higher, something like that. Valuation perspectives here, we had an investment banker come in and do a discounted net present value, implied enterprise value analysis, and it looks like our company is worth somewhere around $7 billion right now, even in the preclinical phase. Now, is that what we're going to get funded at? Almost certainly not. But it's a good argument to start the conversation. Our current raise is we're raising $20 million on a convertible note right now. We have about $8 million committed to that. Conversations are underway right now. And then in early 2023, we'll raise another $100 million. And that is the presentation. Happy to take any questions now.